Thank you. Big round of applause for the participants of this specific session. We will showcase 3 projects that are going on in the Baltics right now. All brilliant ideas and wonderful people. And without further ado, we'll start with the Latvian iGEM team. Please, Kaiva and Alex.
Thank you. So today we'll be taking you through our iGEM journey, our experience, our achievements, and our future prospects, essentially putting Latvia on the map of the largest synthetic biology competition in the world. iGEM was founded at MIT over 20 years ago, and it started off as a small initiative, but it soon grew to be the largest synthetic biology competition in the world. And you can read the statistics on the screen, but one of the biggest takeaways is not only the development of synthetic biology as a field. Many startups that grew to be very successful companies have started in iGEM, but also the community that it fosters and the incentives and interest it sparks for even high school students for synthetic biology. And talking about Baltics, it's a very valuable initiative to bring also here. Estonia and Lithuania have been participating in iGEM for a bit longer than us, and they've gained a lot of recognition and very notable results in the last year's competition. And it's very exciting that Latvia is also participating for the 3rd year in a row already. And more about our journey. Our first project, the team focused on the biosynthesis of crocin, which is the component of saffron. And they used an unconventional yeast to produce this biosynthetically. And for this, they gained a bronze medal, which is a notable achievement for first-time triers. And we could learn a lot from this team and feed this feedback into the next year, where I was part of, and Aleksandrs as well. Last year's project, we focused on cyclic peptides, and these components can be used as antibiotics or pesticides. And what we did is we took the burp domain, which is a plant-derived protein, which can cyclize peptides. And we proved that this protein can, in fact, cyclize linear peptides. And this was our start of our sort of project. And we also focused more on community engagement and education and human practices. So we developed a board game for high school students, which is now turned completely digital. And if there are any teachers in the conference, then you can feel free to reach out and we'll send over the details. And yeah, we iterated this in a few, 5 and more classrooms and took direct feedback from students to make changes to our game. And last year we were awarded the gold medal, which is a very nice upgrade from the first year. And we also got a nomination for Best Basic Genetic Part. And we also got a lot of media recognition, including being on Latvian television and Latvian radio. And
indeed, living iGEM Grand Jamboree as the first gold medalist from Latvia is a life-lasting experience and memory. But in fact, our journey did not end in Paris. In fact, right after it, we have participated in the EIT Food Challenge Labs 2023 in cooperation with BioCrop Science and also Dojus Latvia. And it was a 3-week-long experience when we tried to tackle modern agricultural problems by implementing our scientific work as a way to produce new, biodegradable, effective pesticides based on the cyclic peptides. And as a culmination of this event, we were flattered and honored to win in the category of advanced crop protection. But I would like to also give you a short sneak peek into what our team is working on this year. So we have decided to approach the problem of hypoxia, which is a state of oxygen deficiency in tissue. And in cancer, it is closely associated with tumor progression and poor response to therapy. And it is really hard to detect and measure it. So our goal is to develop a biosensor based on the HIF signaling pathway that would be capable of doing both. To conclude this presentation, I would like to just highlight how valuable and life-changing experience of participating in iGEM was for me. It is a great opportunity to develop both scientific research and soft skills. And also, I would like to thank Biocatalyst Foundation and Andrii personally for kickstarting our team 2 years ago. If you're interested in our journey, please follow it in our media. Thank you.
Thank you so much. They say that iGEM is like a startup factory, and in fact, the biggest synthetic biology startup or company in the world right now was born in iGEM 20 years ago. So there's more to come, hopefully also from Latvia. And now we zoom out to the Baltic context. One great initiative that has been going on in the Baltics for the last months is called Nucleate. It originates from Boston, and now it's also in Tallinn, Riga, and Vilnius. And we have here today with us the winners of the first Nucleate Catalyst program. Please welcome Filips and Christopher from ReOcto.
Hello. Can you hear me? Yeah. Hi, everyone. I'm Philip from Team ReOcto, and this is Christopher with ReOcto in his hands. So we're making a small and smart bioreactor. And as it is biotech stage, I think I don't have to persuade anyone that biotech holds the future. However, there are some bottlenecks in this field. And one of them is developing fermentations. And this is what we want to tackle with ReOcto, giving scientists a tool to study fermentations in an easier and more affordable way. So, will there be a presentation?
There might be a technical problem with this one. Fortunately, have a wonderful prop. This is how it sometimes is with startups, you know, so things don't work.
Let's go. So if you're developing a biotech product, most likely you start by finding out which strains to work with. And for this, you start with a small scale, so tube and maybe a bigger tube. And exactly, this is how you confirm that your organism is alive and that it produces something which is similar to what you wanted to produce. Next stage, you go to flasks. A bigger volume, you produce more biomass, and you can confirm your findings. And usually this stage is quite straightforward. You don't expect many surprises there. However, and also, this is sometimes here everything stops the development, and you just use flasks for production of your organism because you can get enough biomass. However, now, more and more slides are appearing, so we're at the stage of confirmation now. Here, right, when we go from one strain to one smaller biomass to a bigger biomass in flasks, and then we need to go to bioreactors in many projects where you want to already work on industrially relevant things. And while the first step, as I mentioned, is quite easy, the going from flask to bioreactor can bring a lot of surprises. So this is where we come with ReOcto. We want to combine advantages of the flask, so cheap and easy to use, let's say relatively cheap, and of bioreactor that allows you to control multiple parameters and also monitor growth of your organism online. So we developed this product which can control most of the important parameters of the fermentation, such as temperature control, online optical density measurement to check the growth of organism, adding some substrate, base for modulation of the environment. And we also developed software that can control this reactor remotely, so you can access it from anywhere easily. If you are a microbiologist or molecular biologist who have never worked with reactors, but you're working with flasks, ReOcto can help you and serve as a entry point into bioreactor world, where most likely at some point you will need to go, and it allows you to do it simpler, taking less space and a fraction of the cost. If you're already working with reactors, then still ReOcto can bring value to you, because usually you still use Flask and then you go to the reactor. And ReOcto would be a convenient link that would allow you to check strains before they go to the reactor, thus saving you quite a lot of resources and time as well, so that your success rates of fermentation tests would increase significantly. So we are a team of 3 biotechnologists, but also with a background in sales and product development, and we encountered these problems of fermentations ourselves, so we know what scientists need, and we also see how we can deliver a solution for this. So thank you for your attention. This is, for your entertainment, a picture of Saccharomyces cerevisiae that we fermented in our reactor, and if you think that our product in anyhow could help your projects, please reach out to us. We would be very happy to talk. We are planning to launch already sales in the, let's say, end of summer. So we are very open for talks. Thank you.
Thank you. You can catch Filips or Christopher throughout the day and actually have an opportunity to explore the prototype in real time. Last but not least, we have Aigars, lead researcher at the Riga Technical University Water and Biotechnology Institute and co-founder of the algae phosphorics startup. The floor is yours.
All right. So every year we generate huge amounts of wastewaters, and despite the very efficient wastewater treatment that has been used, it still falls back at recovering resources such as water, nutrients, and energy. Instead, these resources are released back into the nature and exacerbating environmental problems. And since we have to deal with more complex pollutants and more frequent water shortages, it has become really a necessity to develop more advanced and targeted water management solutions. Also, it has been projected that water security will become the global target for investments over the next decades. So the global economy, it heavily relies on water, but water resources are becoming less dependable due to factors such as climate change, demographic shifts, and overexploitation of natural resources. Although there are many advanced wastewater treatment technologies available, they are often rejected due to their rather complex nature and also financial obstacles. On the other hand, microalgae cultivation is emerging as a promising technology. Not only it removes pollutants from wastewaters, but also transforms them into valuable raw material that can be used for high-value substance production. But another obstacle remains here, which is that wastewater-derived biomass is often stigmatized and rejected due to contamination concerns. And this is where the algae phosphorics story begins. It all started as my PhD project at the Riga Technical University, where I developed a solution for municipal wastewater post-treatment using manipulated microalgae that would remove phosphorus and recover phosphorus at an accelerated rate. And this solution showed remarkable results for phosphorus removal. Well, the whole phosphorus content in wastewaters was reduced in just a matter of few hours, while simultaneously producing abundance of proteins. But algae has much more greater potential. As I said, it can be used as a valuable biomass for animal feed, bioenergy, food supplements, pharmaceuticals, cosmetics, natural colorants, biostimulants, and so on. And to address the concern about possible contamination in the biomass, we are focusing this technology on more predictable wastewaters from food industry, more specifically breweries, dairy production, fish farming, and hydroponic farms, for instance. And also the wastewater treatment for food industry is becoming more demanded, and well, it's a growing business sector. And over the course of development, algae Phosphorex has been involved in different support programs for deep tech startups such as Commercialization Reactor, City2C, and most recently Future Hub Climate, which all have provided invaluable knowledge on how to manage a startup. But as I said, our site is set to the food industry. Therefore, I am very excited to share with you that algae Phosphorex has been approved in the EIT Food Seedbed Incubator Program, where we will try to validate our business model and market fit. But our ambition goes far beyond business validation. Our goal is to develop a resource recovery plant that would purify food industry wastewaters, recover resources, and return them back into the production process. And to achieve this goal, we are actively seeking opportunities to raise funding, of course. And if this project has caught your attention, then don't be shy to connect with us and let's have a chat. We will be somewhere around here. Thank you.
To all the big round of applause to all the speakers. Innocent? Yeah.
Thank you, Andrii, for introducing the young talents and potential businesses on the stage. And I wish you all the best in your effort and in your development ambitions. May you succeed, and we'll see you back with already like successful cases for mature businesses.